nucleotide is missing or one nucleotide is extra added. If you have changes in DNA that span more than one nucleotide sequence, that will be considered chromosomal abnormality. So you have chromosomal abnormalities, which you also have structural and numerical abnormalities. In the numerical abnormalities, there is a possibility, in structural abnormality, there is a possibility that you have changes, deletion, or addition of big portions of DNA, extra large pieces or fragments are either missing or added. So anything that could cause mutation is considered mutagen, not only chemicals. This lecture was prepared from what has been sent by medical education department, so I have no, let's say, basically no addition to the lecture. So it's considered to be was from Kufa University or whatever Lister University. Because they don't like changes anyway. So in their definition, they say mutagen is a chemical that causes mutation. I would say a mutagen can be a chemical or a physical agent, or even a biological agent, like viruses. Some viruses can cause mutation in human DNA. So mutations can result from errors that happen during DNA replication. If they are not repairable, they can result from extrinsic factors, exposure to ionizing radiation, UV, X-ray, etc. It can be caused by viral infections, it can be caused by drugs, by chemicals, lots of things. Sometimes it's considered to be spontaneous. It just happens. Why is that? When we say spontaneous, the whoever is not spontaneous if you think about it. In cytosol of every single cell, most chemical reactions that occur, there is a liberation or release of what we call a free radical or reactive oxygen species. These free radicals carry either negative charges or they have unshared electrons in their orbits. This damages genetic material, DNA or RNA, and sometimes even proteins. So when we say spontaneous, it means within the cell because of the byproduct of chemical reactions during metabolism. You can define mutation in another way. We know that in 2001, two respectable journals published the first draft of Human Genome Project. That was considered one of the big milestones happened in the history of medicine and biology. At that time, about 90% of human genome information was revealed. Just recently, a few months back, the other 10% of Human Genome Project was completed. So think about from 2001 until 2022, the Human Genome Project has accomplished. Based on certain criteria, randomly from certain people around the world, people who were considered to be normal, healthy, not having any chronic diseases, not having any genetic diseases, they were actually selected at random, not including any people from the Middle East, unfortunately. You need to know that for sure. All from Canada, Australia, Europe, and America, and that's North America. So these people, around 1,000 people, at random, samples of blood were taken from them. Anyway, the brief of speech is that the Human Genome Project was based on samples from people representing population of the world, let's say just for the time being. That genome project, when it was finished, the first draft, so there you can be, was considered to be the reference genome. So we consider it as normal or reference genome. So any deviation, any change, you compare it with that reference genome, and it's not considered to be the same, that's mutation. So this is not an absolute speech, it's relative, you can argue about it, there's no doubt about that. Mutation might or might not change the genetic codon as we we know it. And it may ultimately result in the change of the intermediate molecule mRNA and the protein in case of protein coding genes. These are some types of mutations that were put in the notebook: substitution, deletion, insertion, and rearrangement of our chromosomes. I have a better classification for that, much simpler. You can say you have point mutation, you have chromosomal cross mutation. Chromosome is at chromosomal level, point is at the DNA level. And for that point mutation, you can have silent, missense, nonsense mutation, frame shift, insertion, deletion, etc. So that would be a better classification for mutation. An individual within a population having all the features of a normal phenotype is different from what is considered mutant. So what probably in gene inheritance we learned about these characters, mutant, wild type, allele, etc. The spontaneous mutations, which I consider to be caused by products of metabolism. There is some good news about that and some bad news. People can survive in life without having any diseases, having accumulated even few mutations, more than a few mutations, spontaneously. You like it or not, if we do a survey, a random sampling from people or from among us, even including myself, we could find mutation in certain genes, maybe a few mutations. And we may spend a normal life without having any problems. This is the spontaneous mutation can be repaired by the repair system. You remember when we talked about DNA replication, there is something that's happening after DNA replication, which is repair. If DNA gets damaged somehow, it gets repaired. If you have error during DNA replication, it will be repaired. So we have DNA replication machinery, we have DNA repair machinery. Most of the proteins and enzymes in DNA replication machinery are participants in the DNA repair system. Spontaneous mutations, most of the time they get repaired. They happen, they get repaired. They happen, they repair. If the repair system fails, then you get disease. You get some abnormalities or accumulation of these mutations and then a pathology. Induced mutation is by mutagens. It can be by viruses, can be by exposure to UV light, can be by chemicals, drugs, exposure to any chemical, physical, or biological agent. Does it matter to be in somatic cells or germline cells? It does matter. If you have mutation in somatic cells, you may get away with that mutation without having any problem again. Somatic cells have, maybe a single cell have mutation, but that doesn't mean that mutation rule applies to the whole organism. But if you have germline mutation, then that germline comes from egg and sperm as well. So that they carry the mutation, and in the offspring individual, you have the same mutation in all the resulting somatic cells from that germline mutation. So germline mutation may result in an individual offspring that all of the cells carry mutation, which often result in an individual having a genetic disease. We have lots of these examples: single gene and multi-gene diseases, cystic fibrosis, for example, sickle cell anemia, achondroplasia, scleroderma pigmentosa, von Hippel-Lindau syndrome, lots of them. Okay? There's a question that bugs people in the scientific community, and they keep asking about this. Anyone who studies mutation, this question comes to your mind. Is mutation beneficial in human? We don't have such a thing as a fixed fact. Mutation can be, look, because mutation, the changes in DNA, the mutation that happens is random. For that reason, it's almost always considered to be deleterious, detrimental, bad. It's not a good thing. It's good for other organisms like bacteria, which are prokaryotes. Bacteria can acquire resistance to antibiotics by having mutation in one gene or more than one gene. In human, talking about human, is mutation beneficial? I would say maybe there are examples of beneficial mutations in humans, and I will give you some of these examples. I didn't put it there, but these examples doesn't mean that the individual is totally healthy. Maybe they are suffering from some other diseases, or they have a genetic syndrome, or they have something in the background, a disease, a pathology, but that disease situation made them benefit from something. For example, we know that sickle cell anemia is caused by changing one base pair from GAG to GTG, so glutamic acid becomes a variant. Single nucleotide chain from A to T, that type of point mutation makes cells to change their biconcave shape to sickle. What we see there showing Professor Rega. These sickle cell red blood cells are resistant to malaria parasites. None of the other red blood cells that are biconcave are resistant to malaria parasite. So you have a sickle cell anemia on one hand, and on the other hand, there is resistant to malaria parasite. Another example, there is a tribe, Tabilean tribe in Milano, in Italy. They have abnormally high density lipoprotein, apolipoprotein, HDL, which among people is considered a good cholesterol. Abnormally, they have high level of HDL, which people makes them to be less prone to cardiovascular diseases, hypertension, heart attack, myocardial infarction, angina pectoris, etc. So another example is, which I think this needs more study, tetrachromatic vision. But they say in books, millions of years ago, I don't know how long, but humans were unable to see all of the colors. Maybe we were able to just see two colors, red and green, or blue and green, or blue and red, two of them. So the people were dichromatic vision, have dichromatic vision. But now we are having tetrachromatic vision because of mutation. This is another example of beneficial mutation. So I would say a mutation is good for human if it's not affecting your health, if it's not like critical gene, or a mutation can be beneficial if it gives you an advantage to survive better in a harsh environment, regardless of any conditions, by natural selection. يعني مثلا خلق للسوء تاو يستهادفين لجن ولا صعب يجلس ويروفروا أجل mutation يقول يا بيقول تقول survival معناه لإنسان بيبقى وينك هيجلا وكاركان يجينجل دبيتو دافع ودكان شاخص صعوبة يجي هل كارساتك هل كرس واجد بيبقى إنسان هل survive هل منته بشيء the whole will mutational. So mutation may be advantageous if it gives you variation, genetic variation by natural selection in any environment. Anyway, so are mutations good? Mutations are not good or bad per se by themselves, but they are just different. So this is what I wanted to say about that statement. Is mutation beneficial or harmful? You can say it's beneficial for other organisms. It can be harmful most of the time to human, but there are also examples of good mutations in human. And these mutations are associated with other conditions. Somatic versus germline. Any mutation, the somatic cells are somatic cell mutations. So germline includes spermatozoa and ova. have mutation at the DNA level, single nucleotide changes in nucleotide sequence, or sometimes even more than a single base pair. That's point mutation. And based on that, we have silent, missense, nonsense, and sometimes insertion and deletion, which we call angle mutation, friendship. Cross mutation is chromosomal mutation. Mutation affecting larger portions of mutation. Examples of chromosomal mutations, cross mutations, autism is one of them. There are, if you see all the 23 chromosomes in autistic people, you will see parts of chromosomes are deleted on more than one chromosome. Point mutation basically is considered to be either transition or transversion. Transition, purine change, purine or pyridine. So same, within the same, either two ring or one ring. Remember the 4 bases of DNA, adenine, thymine, guanine, cytosine. Transition is purine to pyridine or vice versa. So based on point mutation, the transition or transversion, I'll explain that in a minute. Just bear with me. Point mutations, either base substitution or insertion and deletion. So you have those two types. Remember this, purines to guanine and adenine, 2 rings, and pyrimidines, cytosine, thymine in DNA and RNA in uracil in RNA. So if it's transition, it's point mutation where purine is replaced with another purine. So in this case, adenine is changed with guanine or guanine with adenine. Amjora mutation agar the DNA replication ho rashi pe aur ki razane navi ki hai bas DNA replication ma ke har ek strand ka bawana parent older ba qawl or daughter strand synthesis but being base pairing group replication agar hua if you have a transition or transversion replication mismatch. So one of the types of mutation that happen after DNA replication is mismatch error. Chun cha ke bedam?
Bir sistem mismatch.
System repair system replication penalty MMR. Mismatch repair system. Transversion is point mutation, but pyrimidine to purine or vice versa, purine to pyrimidine. You have 3 types of these point mutations affecting the coding region. Synonymous or silent mutation, morativity and overwires, saying missense and nonsense. In missense, silent mutation, the genetic codon changes, but without change in the amino acid. Example, you have wild type gene change in one of the nucleotides without change in the amino acid. So that's why it's called silent mutation. Here's an example here. What is silent mutation man here? Normal variation, variation. يعني هو كسر كسيجي كنا همان جينتها يجي السود دربانك ولا كده بعض زمان يجي همان دربان مش بوش السود دربانك لبري جينتيك فاريشن يكمان هي من أي سنة طبعا كيف بخطأ دربانك بدراسة جينوم إنسان كمان نقدر أو جورا بيناتشيا كيف جينتيك كودن تيبل degeneracy redundancy. More than one genetic codon codes for the same amino acid. So based on that rule, you can have a silent mutation and you can survive with that silent mutation. So one of the examples why people can have mutation and spend a normal lives. Mutation is there, but you have the same amino acid. In this sense, there is change in the genetic codon and change in the amino acid. Nonsense, it becomes a stop codon. Protein götürürse ve çift yarayın. اگر پروتینێک دەبێت لە جینێک بۆ دروست بێت لە ناکرا و میوتیشنێک لەناو ناوەڕاستی جینەکە بووبات پروتینەکە دروستکردنەکەی کۆتایی نییە premature termination چی پێڵێن پروتینەکە truncated protein it's a pathological protein. Within the coding region of a gene, mutation can be again harmful if you have insertion or deletion within the shift of the genetic code, within the frame of the genetic code, that's frame shift mutation. Yani frame shift. Yani hemen serdiklotayda geldiğim de genetik kod var. Agar bir türlü serdiklotayda yani hemen ne var yani? Yani çekileler. Yani kodona da çiğliye, de tu yarı beçuar. Diklotaydaki ki bu ziyade bakamadı. Ama bir yanın çiğ, yani karar bir frenogam bormuş, frame shift. Yani devar frenogam bor, yani genetik kodona da tekçe. Agar AUG de burada bu vitamin start kodona AUG bu vitamin. Agar devani AUG var diklotaydaki betenav yani يطلق بها أو أعلنها يوالي فإنه كعبارة frame the shift in the frame of the ring. Why is that? Because of insertion or a deletion.
مرجع للسينيكلوتايد زور تربية أجاب من فريم ورك الشيفتر
أبيت طبعا إذا نيكلوتايد زيادة أبيت اشتباه لأنه يجب أن تطبق وجهك That's mutation in the coding region. Both when the coding region again. Capacity, when we talk about silent, missense, non-sense mutation, why we mention coding region? Because it's happening in the coding region of the gene. So we are waiting for a protein to be produced. Is this protein normal or abnormal? But mutation can also happen outside the coding region. In that case, I want to call it silent, missense, non-sense. أنا الامتحان بسير جوات يعني زول أصلا. قالوا missense, silent, nonsense mutation. Bu coding region. Şunu çağırırken de mRNA protein coding region. But قالوا mutation can also happen outside the coding region. يعني لأنه no coding region again. It's either upstream of the gene or downstream of the gene. In other words, it's in the regulatory sequences of the gene, in the DNA that comes before or after the gene. In that case, I won't be needing silent sense, non-sense mutation. There's no such a thing because you don't have an mRNA resulting from the sequences outside the coding region. But you can all have insertion and deletion mutation. Does that make any sense to you?
شو base substitution إيش هربوا ما اشتبه insertion deletion
substitution or insertion. Base substitution is insertion. Sorry, but base substitution is insertion, but it can be substitution can be changed like mismatch.
affecting non-coding إيش قاعد coding إيش
Yes, also can be a mismatch. اگر تو بس mRNA کلیک به پروتین کدینگ چینه mRNA رو چهار که چی دیگه پروتین چون که کدینگ ریجنت یعنی همون mRNA که هر سری پروتئینی به کدون نگشتن داره اگر کدون نگه بگوارد برای پروتین نگه نگوارد آوا سایل میوتیشن اگر کدون نگه بگوارد پروتین نگه شده که فوره مانوس دیگه آوا میسنس اگر پروتین که کدون نگه بگوارد برای پروتین نگه علاوه مانوس دیگه بسیار کدون آوا میسنس میوتیشن آوا کدین ریجن راست است از اگر باسی سیکونس کم DNA که پیش جینه که بید یا زور بدوی جینه که آوا حتی شده کنی نه کدین ریجن بید طبعا بیارم regulated sequence mutation هتی عربه اندازه mutation که silent missense nonsense نیم بیارم insertion yani deletion yani bunun معناه باشه بشینان frame shift چونکە فریم شفتی فریم خوێندنەوە کێیە؟ فریم خوێندنەوە کۆدۆرە. Frame of reading is for genetic codon. Insertion or deletion. Insertion frame shift mutation. Deletion frame shift mutation. That's very like the coding region. Outside the coding region, I call it just subtraction or addition. Insertion, deletion, mutation. There is no shift, change in the shift or change in the frame of the region. I think I'm coming to the very end of this talk. Repair can happen if you have mutation, especially this content. Even induced mutations can get repaired by DNA repair system. We have lots of examples of genetic repair system. Just one of them is mentioned, I think, here. Mismatch repair. Okay, because حمد هذا بعد شوي مكان بس نوعكان شان ده يعني مطلوب يعني ده أو كده مثلا مكين إذا بس مقطع وتعلم بس توزع بس ريبيرنا فويواني على راسه يعني زود ما هي لو ريبيرس اگر کشای رو برایش DNA replication او کاتا DNA که چون حاصل کرد با اسپیس یه زنه امروز سایکل منگوه پیش اسپیس چی معنی؟ گپ وان G1 فیس یا G1 فیس ها چک پوینت معنی پیش روی DNA که چطور نو اسپیس رو سنتسیس همون DNA که سکانه کرد or damage or mutation. Again, mutation and damage chocolate or DNA space or DNA synthesis and duplication. That's why space is G2 here, G2 face here, G2M here. Before mitosis, there's checkpoint. Now, again, how to DNA replication and mutation and show. That's why our at one level is the chocolate or repair system magnet. چون DNA damage there, actually DNA albocatic kimyayı ve fiziğe ve biyoloji repair system is there to dynamic flow. And you have DNA damage, but you also have DNA repair system.